I'm Judith Campisi, and I'm at the Buck Institute for Research on Aging in the Lawrence Berkeley National Lab. And I work on aging. This is my disclosures. And I want to start by talking about aging and the many diseases that we tend to acquire as we get old. And you'll notice that they're very different tissues, very different manifestations. And yet, what they all have in common is they're very rare in young people, and around the midpoint of our lifespan, so 50 to 60 years of age, in the case of humans, they all rise with approximately exponential kinetics. So those of us who work on aging are convinced this is not a coincidence. We think that there must be some basic aging processes that are driving all of these diseases. And I'm going to tell you today about one process called cellular senescence, which is a candidate for being such a basic aging process. So what is cellular senescence? Well, it's a state. It's a new state that cells enter into. It has three parts. It entails an irreversible arrest of cell proliferation, somewhat resistance to cell death, and a very complex multifactorial secretory phenotype called the SASP, or senescence-associated secretory phenotype, which we think is important for many of the manifestations of aging. So why would a cell enter this state? Well, one reason is because of stress. And many of those stresses that will drive a cell into this state are stresses that are known to cause aging as well as cancer. And so you can see I've listed some of them on the bottom. But there's another way that we have to think about this very complex phenotype. And that is that it was under evolutionary pressure to evolve. And that evolutionary pressure had a number of endpoints. One was this growth arrest, this inability to proliferate, turns out to be very important for keeping cancer at bay in younger people. And cancer is, in fact, very rare in young people. Also, the secretory phenotype is important for fine-tuning certain structures during embryogenesis, and also for initiating labor at the beginning of parturition. And then finally, I'll show you a little data that the secretory phenotype is also important for wound healing and tissue repair. So the way we really need to think about this process is that it is an evolutionary balancing act. That is, it was under selective pressure for keeping young people healthy. And the downside is that as we get older, it begins to show manifestations that we recognize as aging. So let me show you some of the data that suggests this is true. And it starts with the definition of senescent cells. Unfortunately, there are no senescence-specific biomarkers. There are a number of markers that senescent cells frequently show, and I've listed many of them here. And these markers are, in fact, often clustered together within a single cell. But again, none of them are really senescence-specific. So we have to go by clusters and tables of senescent markers rather than any single one. Nonetheless, using these markers, many labs have asked the question, well, when and where do you find senescent cells? And the answer is twofold. First, they increase with age. And this is true for virtually every vertebrate species that has been examined, and also every tissue that has been examined. And the second place you see them is at sites of pathology. So if you compare, for example, an osteoarthritic joint with an aged, matched, non-diseased joint, there are more senescent cells in the diseased joint, and that goes true for a number of age-related pathologies. So this says, in a way, the cells are present at the right time and the right place to be a basic aging process. But here's another problem. The cells are never abundant. At the most, you might see a few percent of senescent cells, but they are never extremely abundant. So given that they're present at the right time and the right place, how could they possibly drive so many different pathologies, given that they're so rare? And then, of course, the big question is, do they drive these pathologies? Because everything I've told you so far has been correlation. So let's start with how. We think we understand how, and it has to do with this very complicated secretory phenotype that have been described by many labs. And what the secretory phenotype entails is many cytokines and growth factors, proteases, lipids, and small molecules. Not all of them can be detected with single cell RNA-seq, for example, but some of them can. But what they all have in common is they promote inflammation. And inflammation is probably the one hallmark of an aged tissue that everyone can agree on. And many years ago, Claudio Franceschi coined this term, inflammaging, to describe how a pathologist would identify an old tissue and a young tissue. We know that inflammation will destroy tissues, will prevent normal cells, including stem cells, from functioning properly. And it's a great risk factor for the development of cancer. But of course, it takes time. So it's late life cancer. On the other hand, you'll never heal a wound or repair a tissue unless you have at least an initial inflammatory response. And the last point I want to make about this secretory phenotype is that we call it inflammation plus because not everything that the cells secrete are driving inflammation. As I said, certain things like growth factors, for example, are not necessarily driving inflammation. But it is enormously complex and very, very plastic. And let me give you some examples. So first, let me show you what happens if you take a human prostate, same person, so the genotype is the same, and you separate the fibroblasts from the epithelial cells. This is an old experiment that was done by Jean-Philippe Coppe when he was in the lab, and he was using an antibody array. And what you can see is that there are certain, so yellow is high, blue is low. You can see that there are certain compounds that are specific to both the fibroblasts and the epithelium, but there are things that are fibroblast specific and things that are epithelial specific. And now with RNA-seq, we can go back and we can look more in depth, for example, comparing fibroblasts with astrocytes, human. There's about 50% overlap. And there are species-specific differences. The mouse is still our best model, but unfortunately, there are differences between mice and humans. The other thing about this secretory phenotype is it's not static. It changes with age and it changes with time. So you're looking here at the increase in gene expression of two lipids. One is a leukotriene, and that is pro-fibrotic. This is ALOX5. And the other is an anti-fibrotic prostaglandin. And you can see that if you induce, these are normal human lung fibroblasts, if you induce them to become senescent, in this case, by DNA damage using irradiation, you can see they have very different time courses. And this is important because we also compared wild type human lung fibroblasts with human lung fibroblasts from patients with idiopathic pulmonary fibrosis. So again, you induce senescence, and the profibrotic leukotriene is induced just fine. But you'll notice that the anti-fibrotic prostaglandin is deficient in the patient population. And we're sure that this has something to do with the pathology, the fibrosis that these patients suffer from. The other interesting thing about the secretory phenotype is it depends on how cells become senescent to begin with. I will point out one of the big unknowns in biology is we don't know what drives cells into senescence during normal natural aging. But what I've listed here are 3 types of stressors that are known to cause cells to become senescent. And then I've color-coded their secretomes. And what you can see is there are things in common, but there are also things that are specific to that particular type of inducer. And then finally, I should point out that in collaboration with Birgit Schilling, a lot of this work was done by Nate Basisti, who now has his own lab at the NIH, but Birgit is our mass spec guru. And we did an in-depth analysis of what the cells were secreting, both as soluble proteins and as exosomes. And you can see that the range is just enormous. There's a core that is common to all of these, but there are also very distinct profiles of secreted proteins depending upon the inducer. And the last point I'll make is that in addition to proteins, the cells secrete lipids. And this is work that was done by Chris Wiley, who now has his own lab at Tufts in Boston. And what you can see is that there are many, many genes that are upregulated, and those genes code for proteins that synthesize these prostaglandins and leukotrienes. And this is a prominent part of the secretome. So this is the complexity of the secretory phenotype, but does it actually do anything? And the answer is it certainly does in a mouse. And let me show you 2 examples. So the first is what you're looking at on the left are mouse mammary epithelial cells growing in a 3-dimensional culture, and they've been given drugs that will induce milk production. And so the red is beta casein. And now if you embed in those cultures, those 3D cultures, non-senescent mammary fibroblasts, they still make beautiful alveoli, lots of milk. That's very different if you embed in them senescent mammary epithelial fibroblasts. So the last example I'll show you has to do with cancer. And this is what happens. You're looking now at tumor volume over time. And this is a pre-malignant breast epithelial cell line injected into the mammary gland of the mouse. So in the absence of fibroblasts, they don't form tumors. But in the presence of senescent fibroblasts, they do form tumors. And this is in an immune competent mouse. And if you look on the right, these are human breast cancer cells grown in an immune incompetent mouse. And you can see in the presence of senescent fibroblasts, those tumors are larger and they're more vascularized, and they will actually metastasize. So the idea is with age, we accumulate senescent cells, and those cells then secrete molecules that can cause neighboring cells to fail to function. And of course, we're all accumulating pre-malignant cells with age, and we would argue that senescent cells can even drive those pre-malignant cells into malignancy. So what do we do about this? Well, there are two strategies that many people have taken. And the first is to stop the cells from secreting. And we actually took this first, and we began to drill down and identify the molecular pathways that drove this secretion. And we've given this up pretty much now for the following reason, that the pathways that drive these secretory phenotypes really are important pathways that you would not want to inhibit in, say, a middle-aged or even a young individual. Things like the DNA repair pathway, things like the mTOR pathway, which is responsible for responding to nutrients and growth factors. So we thought a better way might be if we could find a way to simply get the cells to go away, because they're rare, there are not very many of them. And then if you could get them to go away, you wouldn't even have to have a drug present all the time. You just have to wait till the cells build up again. And so this would require intermittent dosing instead of constant dosing. So the way we did this is we created a transgenic mouse line in which we used one of the genes that's commonly expressed by many senescent cells, although not all, and that is this tumor suppressor P16. And what we did is we engineered the P16 promoter to drive a reporter that allows us to visualize senescent cells by luminescence, a red fluorescent protein so we could sort the cells, and then a killer gene, a viral thymidine kinase that turns the prodrug ganciclovir into a very potent DNA chain terminator, which gets into the mitochondria, fragments the mitochondrial genome, and the cells die. So what I'm showing you here, much of this, all of this mouse work was done in collaboration with Jan Wies at Einstein and Jan Kwimakers at Erasmus University, and much of its characterization was done by Marco De Maria, who now has his own lab in Groningen. So what you can see is that if you've taken a 12-month-old mouse, so that's like a 30, 35-year-old human, the signal is very low, but with time, there's more and more senescent cells. And if we treat the animals with ganciclovir, we can lower that by about 70 to 80%. And on the right is actually visceral fat. Visceral fat is very rich in senescent cells. And again, treating the animals with ganciclovir, we can eliminate 70 to 80% of senescent cells. So we have shared our mice with many, many labs. I've lost track of how many labs. And each lab has then worked on a different age-related disease and asked whether senescent cells either ameliorate the disease or prevent its appearance, or even at times reverses the disease. And I'll show you an example of prevention and reversal. So we're very interested in certain chemotherapies that are used to treat especially children with cancers. And then these children wind up showing premature aging after 20 or so years. And this is also true for some drugs used to treat patients with HIV/AIDS. So you can treat these animals, these mice, with a single dose of these agents, which is what Marco did, and then wait, and they have a burden of senescent cells. And Marco asked, what's the cost of having that burden? And the first thing he looked at was cancer metastasis. So what he did was he took now a cancer cell line, breast cancer cell line, that's marked with a different luciferase. So this is now firefly luciferase, injects it into the mammary gland, the inguinal mammary gland of the mouse. And it's known that these cells metastasize to the lung and the liver. So the experiment is we give the animals breast cancer, we let the tumor develop, then we treat them with this genotoxic agent, in this case, doxorubicin. And we wait just a few days, and then we give them only 5 days of ganciclovir. And then after 2 to 3 weeks, we image them. And what you can see in the control animal, you can see the metastases to the lung and the liver. But in the ganciclovir treated animals, we completely eliminate the ability of the cells to metastasize. These drugs also cause heart failure, and we can measure that in a mouse. This is a collaboration with Simon Meloff's lab at the Buck Institute. So we can measure the ejection fraction and fractional shortening, which goes down after treatment with the doxorubicin. But if we treat with ganciclovir, again, just a few days later, we can eliminate that heart failure. These drugs also cause blood clots, which is easy to measure in an animal. And again, treating with ganciclovir prevents the blood clots. Now, what I want to point out, though, is if we wait too long until after the blood clots have already formed or until after the heart has already failed, we can't reverse it. So this has to be done shortly after the drugs are given. But there was one example of reversal that we did in collaboration with Jennifer Elisif at Johns Hopkins University. And this was done by Oki Jeong, who now has her lab in Korea. So this is an example of injury-induced osteoarthritis. And the pink color, the orange color that you see is the proteoglycan layer that prevents bone from rubbing on bone, which is what causes the pain. And what you can see is that after the surgery, a few weeks after the surgery, you get bone rubbing on bone. But if you treat the animals with ganciclovir, after several weeks, they begin to regrow that proteoglycan layer and they begin to reuse that limb, which they had not been able to do after the ligament was cut. So let me just end by going back to this idea of wound healing. So I told you that senescent cells are important for wound healing. And if you look at the bottom panel, you'll see that what happens is wounds, this is female mice, they heal their wounds faster than males. Within 5 to 6 days, if you make a little punch biopsy on the dorsal flank, they heal that wound pretty fast. But if you eliminate senescent cells from the wound, they're much slower and the wounds are more fibrotic. Now, what Marco was able to do was to sort those cells and show that senescent cells actually make a growth factor called platelet-derived growth factor. It's the AA form, and that's responsible for promoting wound healing. So there's a bit of a conundrum here. I showed you that senescent cells accumulate with age. They certainly do in the skin, but everybody knows wound healing slows with age. So how do we reconcile this? We went to another transgenic mouse model, also made by Simon Meloff, and this was done by Michael Velarde, who has his own lab in the Philippines. In this model, we induce senescence specifically in the skin, but the difference is, and we do this by activating Cree with tamoxifen. But the difference here is the cells are persistent. And when we do that, we see that again, wound healing is retarded. So the idea is that cells are designed to be transiently present in order to promote optimal tissue repair. But with age, they become persistent, and this is what becomes maladaptive. Now, obviously, we all want to go out and get our transgene, right? And that's not going to happen soon. So are there drugs that can mimic these transgenes? And the answer is yes. There are two types. There are drugs called senolytics, which are designed to selectively kill, and selectively is in quotes because it's not all or nothing, but selectively kill senescent cells. And then xenomorphics, which are drugs that selectively suppress certain secretions. there's still a lot of work that needs to be done because there's lots of unknowns. We don't know, for example, how to target senescent cells with either type of drug. We know that we have to preserve the good ones. And again, we have to take into account this remarkable plasticity and dynamic nature. But nonetheless, these drugs are on the horizon. And I think they do hold promise for extending our health span. Of course, everyone wants to know, will they extend our lifespan? And the answer is probably not. That experiment was done by Jan van Dersen at the Mayo Clinic. He made a very similar mouse. And what he showed is that when he treated the animals to remove senescent cells, they had an impressive increase in median lifespan, but no increase in maximum lifespan. And this is another big mystery in aging. We don't really know what sets species-specific lifespan. It can be done in C. elegans, the world record is 10-fold. It can be done a little bit in Drosophila, which is a more complex organism, where maybe it's 2-fold, actually less than 2-fold. And in mice, we can barely get a 10 or 20% increase in maximum lifespan. So there's a lot to be done in order to really understand how these cells are promoting health span and lifespan, and what can be done about actually living longer as opposed to living healthier, which I think is something that is within our grasp. And I'll stop here and thank the many, many people in the lab who have contributed to this work, and a huge number of collaborators. And without these people, none of this would have been possible. Thank you.